Hello guys, I am Shubham from Shubham Tutorials. This video is about DNA polymerases, types and function. So before we start, subscribe to our channel and press the bell icon to get notified whenever the video is out. So let's get started with the video. As we discussed in the previous video, DNA replication occurs by the specific enzyme known as DNA polymerases. So there are types in this polymerases. In prokaryotes, there are different types, and eukaryotes, there are different types. Okay. So in prokaryotes, the types are five: DNA polymerase 1, then polymerase 2, polymerase 3, polymerase 4, and polymerase 5. These are in eukaryotes. Uh, in prokaryotes, right? Basically, structure of the DNA polymerase is same in prokaryotes as well as eukaryotes. So this is a structure of DNA polymerase. Resembles the right hand structure. Okay, so you can see here finger domain, thumb domain, palm domain. So this is basically an enzyme. This bring about DNA replication. Primary catalytic domain is the palm domain. Palm domain is the primary catalytic domain. From this polymerases of types, the third polymerase, polymerase 3, is known as multi-subunit complex. Okay, it requires a lot of factors with it to function. DNA polymerase 3, this is a main primary enzyme for DNA replication. Primary enzyme for DNA replication. So this is a multi-subunit enzyme. It requires lot of subunits. So this is known as holoenzyme. Holoenzyme. DNA polymerase 3 holoenzyme. So this is structure of a DNA polymerase 3 holoenzyme. We can see here these are three core. Three enzymes are associated. So this is a Pol 3, first core, this is the second one, and this is the third one. Okay, these are joined by these other another proteins. So this is a linker. We can say tau proteins. Okay, tau. This is a flexible linker. We can see here these are the linkers here. They are flexible. Okay, they will slide on the DNA strands. Thus, they are flexible. Now, the sliding clamp. This is one subunit of a holoenzyme. Right? This complex, and this is sliding clamp. Okay. So basically, this DNA polymerase 3 is having a high processivity. Thus, it is very important for DNA replication. It is having high processivity. High processivity means more number of nucleotides can be added in less time. In less time, large number of nucleotides are added. Thus, it is having high processivity than the other polymerases. So, DNA polymerase 3 is the important enzyme for DNA replication because it is having a high processivity. Then, what are the functions of other enzymes? All of the other enzymes are having different functions. So, we can see here a chart is there, prokaryote and eukaryote. So, see, this is a prokaryote. We discussed all three core. These are three subunits, right? And holoenzyme 9. Okay, this bring about chromosome replication in bacteria, right? E. coli. E. coli. Then polymerase 2, known as DNA in E. coli. This work is DNA repair. Polymerase 1, RNA primer removal and DNA repair. Okay, RNA primer removal, we know that. Let's see. This is a DNA 3' to 5', right? There will be primer here. Okay, and rest will be DNA synthesized. Like this. Okay, here will be the bonds, right? And this is 5' to 3'. So here are the bonds, right? I will draw here in red only. So this is primer. So primer is 5 prime to 3 prime, right? As well as if there is a second strand here, we will draw a second strand as well. Opposing strand, this is a strand with 5 prime to 3 prime. Then primer will be this side. Okay, this is the primer and rest is a DNA. Rest is a DNA. And here are the bonds in between. So this primer has to be removed. This is what 3 prime to 5 prime. So what we can see, primer is 5 prime to 3 prime. This is the primer. So this enzyme, the polymerase 1 or the enzyme which is important for removal of primer, that is having this activity 5 prime to 3 prime exonuclease, right? Exonuclease means it will cut the nucleic acid from outside. 5 prime to 3 prime exonuclease. So we should remember this, that Pol 1 is a 5 prime to 3 prime exonuclease. Exonuclease. I will just show here with polymerase 1, right? Then the next enzyme, polymerase 4, known as DIN B. Again, that is also having function of DNA repair and translation synthesis. Okay, translation synthesis. This is discussed in repair only. Basically, main important is Pol 3, right? And Pol 1 for primer removal. Rest other enzymes are for DNA repair. Then Pol 5, known as UMUC, UMUD2C, subunit 3, and this is also for translation synthesis. So main enzyme is what? We can say main enzyme is the Pol 3 core and this Pol 3 holoenzyme and Pol 1 for DNA replication. Right? So these are the important for replication. Now in eukaryotes there are 10 to 15 types of RNA polymerases. Here also the main is the polymerase alpha, which is having a function of primer synthesis during DNA replication. So it is a primase basically. Polymerase alpha is a primase. Then the next important eukaryotic DNA polymerase is the polymerase delta and polymerase epsilon. Okay, delta is for lagging strand. Delta is for lagging strand DNA synthesis. Nucleotide and base excision repair, all are important for repair, but this is important for lagging strand. Okay, Okazaki fragments. Then polymerase epsilon, important for leading strand DNA synthesis. Leading strand DNA synthesis. Rest all other enzymes are important for repair. Okay, one more important is polymerase gamma. Polymerase gamma is doing mitochondrial DNA replication and repair. Mitochondrial DNA replication is done by polymerase gamma in eukaryotes. Rest other polymerase beta, polymerase theta, polymerase mu, all these are used for different repair pathways as well as some other functions. But these are most important: polymerase alpha, polymerase delta, polymerase epsilon, polymerase gamma for DNA replication. So that's it for this video. From the next video, we will discuss actual process of DNA replication, first in prokaryotes, then in eukaryotes. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the video is out. Also, follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.